Protein purification can sound straightforward, but choosing the right strategy can mean the difference between a pure, functional protein and a degraded mess. Whether it's maximizing yield, enhancing purity, or minimizing unwanted interactions, understanding the 4 core protein purification techniques can be really helpful. So in this episode, we're looking at 4 common techniques so you can get a general understanding of the purpose and differences between each one. The 4 main approaches to protein purification are affinity purification, ion exchange purification, hydrophobic interaction purification, and size exclusion purification. Each of these methods targets specific properties of proteins to isolate them in pure form. And often you're not just using only one of these techniques, but you're using different ones in a series of steps to purify your protein of interest. So let's start with affinity purification. Affinity purification is one of the most popular techniques because it's incredibly precise. Imagine you're at a party trying to find your friend, and instead of scanning every face, you simply look for someone waving a red flag. That's the essence of affinity purification. Scientists use a similar trick by attaching an affinity tag, which would be like that colored flag, and they attach this to the protein of interest. This tag binds to a molecule that's already immobilized in a column. This binding basically captures your tagged protein while the rest of the cellular components flow through that column. Once the unwanted proteins are washed away, a change in buffer conditions releases the tagged protein, leaving you with a pure sample. The concept is simple but highly effective, making affinity purification a go-to method for many applications. Next up is ion exchange purification, which takes advantage of a protein's unique electrical charge. Proteins are made up of charged amino acids, and their overall charge changes depending on the pH of the solution that they're in. By adjusting the pH, you can make your proteins positively or negatively charged. And while this concept seems pretty simple, the basis of protein charge can get a little complicated. So I'll have some links in the description for resources about how a protein sequence and the pH determines a protein's charge. Here's where it gets cool though. If your protein is positively charged, you'd pass it through a negatively charged column and voila, it binds. If it's negatively charged, you'd use a positively charged column instead. After the protein is securely bound, a high salt solution disrupts these interactions, causing your proteins to be released in purified form. This method is great for when you want to separate proteins based on their charge alone, making it an excellent follow-up to affinity purification. By the way, if you're wanting to learn more about ion exchange chromatography, we have a video that goes into more detail, and we have a video explaining the differences between cation and anion exchange that you might find helpful. So we all know how oil and water don't like to mix. And we can shake up the mixture, but in the end, it all separates back out. That's because oil is hydrophobic, so it doesn't like water. Hydrophobic interaction purification uses the same principle. Proteins have regions that are hydrophilic or water-loving, and they have hydrophobic regions or water-hating regions. By using a high salt buffer, the hydrophobic parts of a protein are encouraged to interact with the hydrophobic column, sticking like oil droplets to the surface. As you gradually reduce the salt concentration, these hydrophobic regions lose their affinity for the column and the protein is eluted. This technique is useful for isolating proteins that have large hydrophobic patches, which are often key to their function or stability. Lastly, there's size exclusion chromatography. This technique doesn't rely on charge or hydrophobic interactions, but instead uses the physical size of the protein. With size exclusion chromatography, proteins pass through a column packed with porous beads. Larger proteins weave around the beads and reach the bottom of the column faster, while smaller proteins end up going through the pores of different beads and take longer to elute. This method is ideal for separating proteins based on size alone. Each of these approaches to protein purification has its strengths and specific uses depending on the properties of the protein you're working with. And again, in some situations, you'll be using multiple approaches in steps in order to get your desired protein separated out from other proteins and molecules. If you want to dive deeper, take a look at the description for more resources. Also, definitely check out goldbio.com for reliable and very affordable reagents for your lab. Plus, we have so many more resources there for you too.